Hello, hello. Hey guys, while we have a captive audience while you guys are getting food, what we're going to do periodically is talk about some of the shared resources through the Cancer Center. And today I'm going to talk about some of the BBSR resources and some new people involved in some of the plans we're looking to push forward. So this will be just a few minutes right before the talk today. So these are the people involved with the BBSR. You can see bioinformatics and kind of the biostatistics group. Two newer people to the group are Andrew Goodspeed. Andrew, can you wave your hand? He's up there. He's going to be helping manage the core. So if you guys have questions or you like anything that I'll show you here, or just questions on analysis or anything like that, please contact Andrew because he's going to be managing the project associated with the core. The other person is Sarah Ferreira. So Sarah, she's over there. She's a new hire who's going to help with bioinformatics analysis, and she'll be working with Andrew and myself and some of the other people in the core. So one of the things I do want to mention is that we're having an educational series. So as you can see, we've had a couple previous ones. I've done something on gene set enrichment analysis. Andrew's done some work in RNA sequencing analysis, and one coming up. So this is a plug for Eric Clamby's analysis of CyTOF data. That's going to be on April 19th. So please be aware of that. Look for the flyers around campus. If you guys have questions on different types of analysis, or if you have things you want to learn more about, single-cell sequencing, I'm sure Neil over here would be happy to talk about some RNA-related stuff, or Jay would be interested in talking about some single-cell sequencing. So anything that you guys would like to know about as a community, please let me know. So one of the things with the core that we're really trying to do is standardize a lot of the workflows, and we've put a lot of work into actually doing that. And one of those I'm going to talk about just briefly, and if you've heard Andrew talk, he's talked about a tool that's been developed with the core, really standardizing some of the RNA sequencing pipelines. And with that, I'm just going to quickly pop out to some of the tools. So the idea now is that when you guys run an RNA sequencing project through the core, what we'll do is actually, instead of supplying with static files, we're going to supply you with a file that links you to your data in an online application that allows you to really work with the data, to try to get you guys really working with your own data rather than just, again, looking at static forms. So what Andrew's been able to do is, this is just an experiment with a bunch of single samples, different cell lines, and different resistance mechanisms that have been derived. You can download the raw. I'm not going to go through all of this because I don't have enough time, but you can download the raw counts, the counts per million, the normalized data. You could do things like principal components analysis just for display of where your samples are falling. You can look at expression of individual genes according to the different groupings you have. And then you can go and do all of and this is being dynamically calculated in the background, so the whole program is running now. So you can see that PCA is dynamically calculated with whatever data you've uploaded. You can do things like expression by group. So if we're looking at KRAS, what does KRAS look like across all of the different cell lines? You can kind of compare those different levels if you're interested in looking in more detail. And then for comparisons, for example, we can say I'm interested in looking at this group versus this group, and you can go through and flip and kind of make as many comparisons as you want. It will display your differential expression. You could do expression by comparison, so looking at that particular gene and whatever comparison you're making. And then it will go into doing different types of pathway analysis. So gene set enrichment and GO-term analysis and things like that. So it's all built within the app. If you've done recent projects with us, you'll know that we can put that all in there and we'll supply this back to you. In the future, this is how we're going to plan on doing things. If you guys have done work through the core previously and you'd like to use the tool, we're happy to upload your data into the tool and allow you guys to go and use that. So let us know if you have questions on that. The last quick thing I'm going to mention is that we're also interested in doing kind of rapid prototyping with different customers or users. So, for example, we had some single-cell RNA sequencing data that, again, will return back in static kind of figures, which you need for the paper. But if you really want to go through and kind of explore how your gene is being expressed across the different clusters that you're making with your analysis, you can do that. So, for example, we can flip between different data sets. It will update according to those data sets, and then we can search for your favorite gene. So KRAS, and it'll display the expression of KRAS in this kind of little cluster group down here. And then you can see that you're getting differential expression, or you're getting the expression plots according to the different clusters that have been previously derived. So this is just a rapid thing we could prototype and allow you guys to kind of search your data. So that's all I want to say. If you have questions on that, contact Andrew Goodspeed, and I'll let Bruce take over now.
So real quick, first off, I want to make a double plug for the bioinformatics, because Andrew sent me a link for some of the data that I'd had previously analyzed. So I played around with that, and it's fantastically powerful. So if even I can do it, anyone can do it. And you really should take advantage of this, because this is a unique aspect of bioinformatics here, is that they're not just running your data, they're innovating, coming up with new platforms. And so now we're going to have Bruce Apple, Anya Belosova, and Todd Pitts give a presentation on organoid and tissue modeling, the program and the Organoid core facility it's already been going on this campus, but we're really hoping to ramp it up. So I'm not going to give them long introductions, since they're already well known. I think all of them have been on this campus at least a decade at this point.
10 years.
10 years. So if you don't know them yet, you should get to know them. And anyway, Bruce, take it away.
All right, thanks, James. And thanks, everyone, for coming. And thanks to the Cancer Center leadership for giving us this opportunity to tell you about this program that we hope will be a benefit to many investigators and trainees across campus. So as James said, there's 3 of us who are going to speak. I'm just going to give you a quick overview of the kind of the motivation for doing this and the program, and I'm going to fill in for Peter Dempsey to tell you just a little bit about the core facility. Probably the more interesting things to you will be presentations from Anya and from Todd, because so much of this is just very complementary to things that are offered by the Gates Center. We thought this would be a good opportunity to have Anya come in and give you an update on the capabilities and opportunities in her biobank and disease modeling core. And then Todd will tell you about some of his work using tumor organoid models. All right. So just to make sure that we're all on the same page here, what is an organoid? It's essentially a 3-dimensional cell culture model. The advantage of this is that to some degree, these organoids can reproduce the cell fate specification and differentiation that occurs in an organism, tissue interactions, and to some degree, organ function. So what this does is, at least again, to some degree, is recreate the complex environments in which cells and tissues interact, which is such an important consideration for things like cell signaling. Okay, so why are we doing this? Why are we trying to expand this on campus here? So from my own perspective, there are really 2 motivations. So from my perspective as a developmental biologist and now head of the section of developmental biology in pediatrics and with an association with Children's Hospital, I know that we need better models of human development and homeostasis and disease. All right, and I doubt that I have to convince you that it's a real challenge to develop more effective cancer therapeutics, and likely one of the limitations that you all face are the limitations of your models of cell culture and of mice. So one of the big motivations for organoids is that you're able to use human tissues and human cells and to create, to some degree, again, some of that 3-dimensional environment in which a tumor is growing. So also from my perspective then as director of the training program in cell biology, stem cells, and development, we need to be able to give our trainees opportunities to learn how to use these new emerging model systems and how to further develop them to make them more competitive for their career progressions. Okay, so our goals, pretty simple, and they align with those motivations. We want to expand the range and the power of the model systems available here on campus. As James said, some of this has already been going on. What we hope to do is now to expand these models to all tissue and organ models. So we want to be able to use these to investigate the basis of human disease and of normal human development, and of course to help develop new therapeutic strategies. To facilitate that, we hope to be able to provide expertise and reagents, advice, methods, cell lines, and specialized imaging resources. Through the core, we anticipate partnering with the Gates Center and with the Department of Engineering and School of Pharmacy, for example, to help develop new therapeutic approaches. And we also aim to provide education and training. Much of this will be through my training program, CSD. We're launching a new 2-credit course on organoids this spring, and we will be expanding that. We'll be providing consultation and hands-on training, workshops, and symposiums. So we're, for example, we anticipate having a symposium in another year, and hopefully we'll have some of the major players in the organoid research in the country here to present at that. Okay, so here's briefly the organization of the program. So this is an extremely grassroots endeavor here. This really is essentially just Peter and me sitting around going, well, we should do this. Let's see if we can get some money and see what happens. And so this is what's happened so far. So we have a steering committee of these individuals, and we may expand on that. And so there's two branches, two components of the larger program. There's the training and education component. I've already mentioned some of these things. And then there's the core facility, and Peter will be directing that. And I'll say just a little bit more on these specifics in a moment. Okay. Okay, so I also want to make note of a pilot project program that we'll be announcing soon that will be available to at least a subset of you. So this is going to be sponsored by my section of developmental biology and with support from the Gates Frontiers Fund. And so we've written the RFA. We'll be sending that out soon. So here's our funding priorities. It's really the idea is to help people move into using new models and new collaborative work, investigations relevant to child health. That's key, okay? And then utilizing stem cell and organoid models. So the key one is child health, so that's going to eliminate some of you. But if you work on pediatric cancers, we'd love to see some ideas coming from you. These are kind of secondary considerations. They don't have to be on stem cell or organoid-based types of proposals. 2 grant categories. We're going to have some grants for $150,000 for 2 years, and the smaller grants for $25,000. I think we've got enough money to do 4 grants of each category. All right. So again, if you do something related to child health, you might keep this in mind. And like I say, we hope to get the RFA out pretty soon. Okay. So what can you do through the organoid core? It's everything you can do with your stem cell and iPSC models, and perhaps a little bit more. All right, so you'll be able to do genome modification. You can do all kinds of profiling. You can do drug screening. And the additional types of things that you can do is really start to model tissue interactions, and even, as indicated here, host pathogen types of interactions. All right, so what about its applications for cancer? I'm really not going to say much about this. This is Peter's list here. And you'll hear more about this from Todd. I just want to point out a couple of things. One is, of course, you can get patient-derived. You can develop patient-derived organoids that hopefully represent the tissue of origin. And I think probably another big factor for you is the heterogeneity, the ability of organoids to represent the intratumor heterogeneity. That's probably a big challenge for you to try to solve the problem of cancer. So you can apply this approach to a large number of different tumor types. And in fact, this has already been done on cancer. There's a variety of different types of organoids that have been made. Everything with the green check or highlighted in green. There are some other things that have been happening on campus too. There are ocular organoids that are being produced in this, the relatively new cell site program in the Department of Ophthalmology, for example. Okay, so, but again, the goal of our program really is to expand this out to all the different tissue types. Okay, so for the core, all right, so if you want to get into the organoid game, where do you start? So the core really is designed, or the intention of the core is to help you get started. All right, and so we'll be, Peter and his team will be there to help you develop a new model, to work out methods, and to give you some ideas about what you need to be able to do to turn a model into an experiment. So it'll be a source. There will be some repository of normal and tumor tissue types there, and you'll have the ability to generate organoid libraries for the purpose of drug screening, for example. All right, so you'll also be able to purchase specialized reagents there, hopefully for at reduced cost. Peter put in subsidized cost there. I don't know where that comes from exactly yet, so I'm not sure how we're going to subsidize it quite yet, but that's the goal. All right, and again, it'll be a repository of for different tissues that you can gain access to. All right, and then also with we got a CERC grant from the School of Medicine to purchase a microscope. So this is a spinning disc microscope. It's fully automated, can image in several different color channels, and it can do automated multi-well imaging. And so this will be a very strong asset for phenotypic analysis of your organoids and for facilitating drug screening. Okay, so where do we hope we're going in the future? Well, we can imagine doing xenografting types of experiments, co-culture sorts of experiments. For my own interest, I'm interested in trying to introduce microglia into brain organoids eventually, for example. Development of microfluidic technologies and specialized matrices and synthetic hydrogels. Christy Anseth is going to do a sabbatical here on campus, so she'll be here for a few months working with Peter to do this for intestinal organoids, but she really hopes to expand that out to a variety of different organoid types. And then eventually, of course, the goal is to be able to produce transplantable tissues for therapies. Okay. So how are we supporting this? So we actually got some buy-in from a number of different entities. So this really all started with the Gallup Transformational Grant. Peter was operating a small core as a piece of the Gallup program. So the Cancer Center has been a very generous supporter. Department of Pediatrics in my section has contributed funds, and also that's part of the Origins of Health and Disease program. The Gates Frontiers Fund is providing resources not only for the pilot project program, but also we're using resources from them to help get the core up and going. Children's Hospital Colorado. I'm hopeful that this will be something that donors will be attracted to. In fact, we already have a bequest, a fairly substantial bequest that will be used for endowment once that matures, as they say, to help support the core. And then, of course, we got the CERC award from the School of Medicine. So we'll operate the core fee for service. There will be charges for consultation training, for the use of the microscope. You can buy your reagents there. And then, but what we're going to really need is people to write things into their grant proposals, incorporating costs into their budgets, and hopefully, if it works out, writing a portion of a PRA salary for someone who works in the core as part of your funding. But I think this could be a really great assistance to center grants, to T32 grants, and hopefully it will make people more competitive for R21 to R01 investigator-driven sorts of awards as well. All right, with that, I'm going to turn it over to Anya. Has a pointer, but this has a pointer as well. Okay.
Okay, so I would like to thank Bruce and the Cancer Center for the opportunity to present the services that we provide as part of the Stem Cell Biobank and Disease Modeling Core. I think my presentation will be particularly useful to people who are using induced pluripotent stem cells to generate organoids. So Stem Cell Biobank and Disease Modeling Core at the Charles C. Gates Center provides services related to reprogramming into induced pluripotent stem cells, or iPS cells, CRISPR-Cas9 mediated gene modifications, mRNA production for transient transfections, as well as long-term stem cell biobanking. So before I go into a little bit more details of what we can offer to you, I would like to give a little bit of introduction into induced pluripotent stem cells. So we all know that human patient cells are a very powerful tool for research. We can use them as a 2D culture, cell culture for drug screening mechanism studies, or we can make organoids, as Bruce mentioned. We also can transplant these cells into immunocompromised animals, generating in vivo models. But we all know that these cells also have their limitations. If we use primary cell lines for modeling, of course, we have limited proliferative capacity of these cells, also limited availability of the cells. Think about cells which are hard to get from the body. It's like neurons or cardiomyocytes. We cannot really get many cells to do much of a research from these. Also, if we're talking about tumor cell lines, they also have their limitation. If you keep growing these cells, they tend to drift away from genetic and growth characteristic of the parent tumor. And also, if you study tumor cells, of course, you are missing these initiation processes, which are very important to elucidate mechanisms of tumorigenesis. So here, induced pluripotent stem cells can help. Back in 2006, Shinya Yamanaka developed a technique where you can collect cells from the patient and reprogram these cells into, can I get it in one?
Yeah, it's very easy.
Right. So you basically can get cells from the patient and reprogram these cells into induced pluripotent stem cells using just transient overexpression of only 4 transcription factors, OCT4, SOX2, KLF4, and c-MYC. And in a way, you get the cells which are almost identical to embryonic stem cells, which you actually derive from the inner cell mass of the blastocyst. So these cells, because they're similar to embryonic stem cells, they can be expanded indefinitely. They can give rise to every somatic cell type in a body, as long as you know how to differentiate these cells, of course. So if you know how to get these cells, then you can, again, use them in a cell culture for organoid formation or for xenografting. Also, these cells are patient-specific, meaning that you preserve all the genetic modifications, basically the genetic background of the patient you want to study, including all the SNPs, everything you can think of, which is very critical if you study cancer. Also, these cells are amenable to gene modifications, and primarily because you can expand them indefinitely, so you can do clonal expansion of your genetically modified cell. And obviously, there is relatively low ethical concern associated with the use of this technology. So, of course, these cells are also applicable for cancer research. Cancer cell lines have been reprogrammed into iPS cells. And basically, these cells can also be, again, differentiated back into different cell types in the body. This will allow you to study the role of cell identity in cancer development, as well as the role of genetic background and epigenetics. Reprogramming into iPS cells preserves very specific genetic abnormalities which are otherwise hard to model, such as all of these chromosomal aberrations. So if you generate iPS cells with the germline mutations, then you can assess the role of this mutation in cancer progression or initiation. as well as you can use iPS cell modeling to see how environment affects tumor formation. So how can we reprogram somatic cells into iPS cells? As I mentioned, technically you need only 4 transcription factors. And there are several methods available which you can use to make induced pluripotent stem cells. And some of them are mentioned here. On top of that, it's a lentiviral method, and the older version is a retroviral method. Of course, this is an integrating approach, where you would introduce your provirus into the genome. Of course, you are dealing with all of the potential genetic modifications as a result of integration. However, we now know that you need the expression of these factors only for very short duration of time, for the reprogramming to happen. After that, the epigenetic stabilizes itself, and pluripotent stem cells become independent of the exogenous factors. They support themselves through the endogenous factors. So meaning that now we have all of these non-integrating approaches which we can use for the generation of iPS cells, such as RNase and dye episomal approaches. And in the core, we are primarily using RNA approach and episomal approach. However, all of these approaches have one big limitation, is the low efficiency of the reprogramming, where it's usually below 1%. So we're talking about the generation of very few iPS cell colonies from 1,000 cells. Of course, if we deal with that, there is always a concern of preselection of certain mutations during the process. So really recently, we published a paper in Nature Communications where we show that we can now increase the reprogramming efficiency to really an unprecedented level. So we focused on messenger RNA-based reprogramming, where we introduce the reprogramming factors via non-integrating, of course, messenger RNA. So in our cocktail, we also did some modifications. We added additional factors. We also added embryonic stem cell-specific mature microRNA mimics. Of course, they're all non-integrating. We now introduce this cocktail of factors every other day. We need maximum 7 transfections, but technically we need about 3 or 4 transfections to have enough iPS cells. So just some representative results. We can actually get now thousands of colonies, iPS colonies, from only like 200 starting cells. It's in a 6-well Foreman dish. So we are talking about the efficiency almost 800%. Of course, it's possible because the cells keep dividing during the reprogramming and generate multiple sister colonies. So this method works very great for fibroblasts, like amazingly well and consistent. And recently, we were able to adapt this method to cells isolated from urine. So we know that the skin biopsy is a relatively invasive procedure, so many patients may resist even to give it. So urine is like a very good alternative source of cells. You need about 50 ml of urine, spin it down, and you have enough cells to achieve your reprogramming. And that's what you can see here. We can adapt our RNA-based approach to get iPS cells within 12 days of initiating the reprogramming. Of course, many researchers on campus also asked us about blood, because many people just collect blood, it's easy, and we know how to do it. Unfortunately, RNA-based reprogramming doesn't work for blood, and I know that lots of commercial entities trying to make it to work. It doesn't work because it's hard to transfect blood cells with RNA. However, what works pretty well, including in our hands, and we recently adapted this method, is episomal reprogramming. So in this case, we need only about 5 ml of blood. We collect PBMC fraction, and then we introduce episomal plasmids, which encode the Yamanaka's factors. And again, within 2-3 weeks, we have quite enough iPS cell colonies to do any subsequent applications. And technically, iPS cells, which you generate using this approach, are also non-integrating because these plasmids slowly dilute out from the culture about 10 passages, and they're all gone from the iPS cells. And also, these iPS cells are almost identical to iPS cells generated by any other method. So in addition to reprogramming, of course, we do all the necessary characterization to confirm the identity of these cells. As I mentioned, because iPS cells can be expanded indefinitely, we can try modify them on a genetic level. And here we use Cas9 system. So I will not go into many details about the Cas9. You probably all know how it all works. But like very briefly, we can generate like guide RNA, which basically direct Cas9 nuclease to cut almost basically most of the genomic DNA in the predetermined spaces. And once the cut happens, then if non-homologous end joining come into play, then this can introduce indel mutations, which create premature stop codon, and as a result, disruption of the gene or knockout. However, if we provide the correcting template for the correction with the homology arms around the break, then we can then achieve either precise correction or introduction of the mutation we want, or even knock-ins of different markers if we're thinking about that. So in order to deliver Cas9, we also use messenger RNA, and I have to note that we synthesize messenger RNA as part of the core services from the DNA template. Basically, it's just in vitro transcription. So just very briefly, some examples. We showed that our Cas9 delivered as mRNA can basically cut different loci in the genome, and that basically allows us to achieve very precise correction of mutations or reintroduction of the mutation we would like to study. And basically, Sean, Marina, and Kyle, they were involved in generating these data. However, I think what would be of interest to the Organoid program is the generation of lineage tracing model, where we can actually knock in different fluorescent markers and a specific loci, and this will allow us to trace cells which differentiate from iPS cells, as well as actually trace the whole differentiation process. So the first goal for us was, it was to generate constitutive fluorescent iPS cell lines. And to achieve that, it's technically relatively simple. Of course, it's all labor-intensive process. We just knock in this construct into a safe harbor AVS1 locus. So in this case, this construct has GFP here and 2A peptide. So in a way, we can see the fluorescence, and AVS is always active, whether it's iPS cells or cells differentiated from the iPS cells. So in a way, it's like a good locus to put the fluorescent markers. Think about like Rosa locus in the mouse. So, and these data were generated by the manager of my core, Dr. Sean McGrath. And you could see that with the help of the intern who now works in the lab, Macra. So you could see that our iPS cells, they fluoresce green as expected. But again, we're not limited to GFP here. As you'll see later in my presentation, we have actually a wide range of fluorescent markers we can put into iPS cells. And as an example, we actually generated iPS cells with nuclear localized TD tomato, as you could see, as well as membrane-bound TD tomato. And as I mentioned, there are other markers available if people are interested. So, of course, the question next is, yes, we have these fluorescent iPS cell lines, whether the fluorescence maintains once we differentiate these cells into subsequent tissues, in like somatic tissues. One example is endothelial cell differentiation. This work was done by Sinead McGarvey in the lab. So basically, endothelial cells are very important because it's part of the vasculature. We know that there are interactions with vasculature and cancer progression. So, but again, endpoint here is that once you differentiate your iPS cells, the GFP maintains. So in this case, it was wasabi, but it's very similar to GFP in terms of fluorescence. So, and once, okay, endothelial cells are completely differentiated. They continue to fluoresce green. We did flow cytometry, 100% green, and also marker of endothelial cells. We also do some functional tests where these cells can form like capillary-like structures in matrigel. They maintain it, and they basically continue to fluoresce green. So as another application, we actually tried to generate human intestinal organoids, and here I actually was lucky because my manager, again, Dr. McGrath, came from the lab of Jim Wells. So basically, he has very broad experience in generating organoids. So he applied a standard protocol for generating intestinal organoids where you get definitive endoderm first with some mesenchymal cells here present. You see in red, and they're very important because they kind of create the necessary structure for the organoid. As you could see here on the spheroid, they go outside, and eventually they can differentiate into smooth muscles as well as fibroblasts. So what Sean did, he used, again, two different iPS cell lines with nuclear localized TD tomato or membrane-bound TD tomato. Then he applied the factors, activin A and all other factors, to kind of induce this gut tube formation. So once he achieved that, that's how the bright field would look like. And that's basically, it shows the fluorescence is still there. So, oops, a bit slow. Oops. I'm sorry, technical glitch. Okay. Yeah, so you could see all of the buds are formed here. It's basically this early stage, and then eventually you collect these gut tubes and immerse them into matrigel. At this point, you can actually combine different cell types and to see if other cell types can integrate into the organoid, or maybe you can use iPS cells labeled with a different fluorescent marker and to see how they integrate and become the part of more mature organoids, which would look like that. And again, if we go for the fluorescence, lights. Oh. Okay, yeah, so you see it here. Hopefully this forgets it shows. Okay, so basically you could see that the fluorescence is maintained. Okay, so of course the next step is whether we can do knock-in into other loci, so we can really look at gene-specific knock-ins. So this work is still in progress. We just started, but we have very good preliminary data. We basically here attempted knock-in into DNA methyltransferase locus, allele which is actually active in pluripotent stem cells, but it goes down with differentiation. However, it can reactivate during cancer formation. So for that, we use, again, 2A and wasabi, and we did knock-in into the last exon of this allele. In this case, we preserve the functionality of this allele, and at the same time, we see the fluorescence once it's active. So our data show that, again, this is iPS cells. So usually iPS cells, they form very tight colonies. Once they differentiate, the colony starts to disintegrate. So here you could see a little bit of spontaneous differentiation on the colony. And while the main colony fluoresces green, the differentiated part does not, which actually suggests that we achieve the precise knock-in. So once we have pluripotent stem cells, the marker is there. Differentiation triggers the silencing, as we see here. And now we're trying to actually knock in into other genes so we can trace differentiation. So, and it's just to confirm that we have DNT3B active, we have 2A, and well as wasabi in our cells. So in addition to reprogramming and to generation of knock-ins, we can also offer mRNA libraries. So as I mentioned, we synthesize mRNA, so we can do different variants of Cas9, we can do different resistance markers. So in this case, we do very precise, very transient transfection. And this is a list of fluorescent markers which we can offer, and each of these markers, if necessary, can be knock-in into iPS cells, basically giving you lots of flexibility to trace these cells in organoids and potentially in vivo. So just summary, Stem Cell Biobank offers RNA-based and episomal reprogramming of somatic cells into iPS cells, patient cell isolation reprogramming. We use fibroblasts, renal cells, amniotic fluid-derived cells, as well as blood cells. We do all of the characterizations. We also perform CRISPR-Cas9 gene editing, as I mentioned. We do knockouts, we do knock-ins, we do specific gene corrections if necessary. We also help with experimental design. We can help with the design of guide RNA if necessary. So also we offer mRNA library and custom manufacturing. So if you have any factors you would like to express using mRNA, yes, please contact us, we'll help you. And as well as we do some long-term biobanking if you are interested. So at the end, yes, I would like to thank people who are involved in the core, Igor, Sean, Nicole, Kyle, Michael, Amy, as well as extended team members who occasionally jump in and help us, like Sinead with her endothelial differentiation protocol, Megana, Marina, Charlie, and Makra. And of course, I would like to thank Charles Seagate Center for all of the support the center provides for the core. Okay, thank you. So I think, yeah, I'll give, okay, so sometimes it's glitching. Yes.
All right. So what I'm going to be talking about is some work that Peter and I have done as a collaboration. It all started a couple years ago when we approached Peter to help us make some organoids from our patient-derived xenograft bank that consists of colorectal cancer, pancreatic cancer mostly, and some other GI cancers. And with that being, once we were able to construct those, we were able to apply for some pilot grants just to get some preliminary data. And currently we have one from the Developmental Therapeutics Program in colorectal cancer. We have one from that's for esophageal cancer to make organoids. And then we have one from the Wings of Hope for pancreatic cancer research. And so this one is basically to examine Wnt signaling as a potential target, a therapeutic target in pancreatic ductal adenocarcinoma using a patient-derived tumor organoid library. So why PDAC? So pancreatic cancer is one of the deadliest. The 5-year survival rate is terrible. Usually presents in an advanced stage, and it's refractory to most therapies. And a lot of that is due to either inherent or acquired resistance. So a major clinical goal is to subclassify these PDACs into different categories so that we can try to find more effective treatments. So pancreatic cancer has some of the common genetic mutations. About 90%, 95% are KRAS mutant. Depending on where you look, 50, 70% are P53, and then they have SMAD4 and CDK mutations as well. Gene expression studies have been difficult because there's not very much tumor cells in the actual tumor. It's a lot of stroma, and a lot of the stroma is fibrotic. But the gene expression studies that have been done have been able to classify them into either the classical or the basal-like quasi-mesenchymal, where the classical has a much better prognosis than the basal-like. So these organoids will provide us with a new research tool to kind of bridge that gap between the cancer genotypes and the biological phenotypes. So we've been able to get organoids from various places, small samples like from FNAs, larger surgical specimens, even from ascites. They basically just get digested with enzymes, put in a small amount of matrix gel, and then into this human pancreatic stem cell media that has all these goodies in it. So PDAC, the organoids, have various morphologies. These are actually the first 4 that we created through Peter, through the core. You can see they have the different morphologies, different magnifications. They maintain these morphologies throughout. So when they're first created, they have these morphologies, and you passage, and they maintain this. So we've been able to create them from about 80 to 90% of our tumors. So this dramatically increases the biological diversity of the available to study. We see both classical and basal-like subtypes. They maintain the histological, molecular, mutational profiles of the parental tumor. And since they only contain only contain tumor cells, they're amenable to more comprehensive functional genomics. And so for me, being in medical oncology, part of the developmental therapeutics program, we're interested more in drug responsiveness, so we believe that these can be readily assessed with these organoids. Excuse me. So a paper that came out last year by Sato's group in Japan, they were able to determine or take a larger library. I believe they had 39 organoids, and were able to subclassify them by their Wnt niche requirements. So they have some that require exogenous Wnt and arspondin. They have some that require exogenous arspondin, but they produce their own Wnt. And then there's some that they classify as Wnt arspondin independent because they make them themselves. So this is completely autocrine. And so they determined that these biological phenotypes could not be predicted by genomic or transcriptional profiling. And the reason this is important is because as you become more Wnt independent, it increases the metastatic potential of these tumors. So for us, we started this because we had a fairly large patient-derived xenograft bank that consists of colorectal cancer and pancreatic cancer. Now we have roughly 18 models growing in mice. We excised them, basically just gave them over to Peter. A lot of this work was done by Betty Jakob in the lab. And we created a bank of PDAC organoids. If we have enough tumor from the patient, we also take patient tumor directly and make organoids. But our first priority is our bank to get the tumors growing in mice. We've done some targeted sequencing on these through Komoko, and they basically, the story is they maintain the mutational profiles of the parental tumor and the global pancreatic cancer mutational profile. So what we decided to do is we're for this grant, it's to take all of our pancreatic organoids and classify them by their Wnt dependency based on that paper from Sato's group. So here's just one example of Panc 269. This is the tumor or the organoid growing in the complete media. You can see everything down here. And basically growth factor withdrawal until we get down to just media alone. And you can see that with this model, basal media alone, the tumor doesn't grow, but all it really needs is our spondin to grow. So what this tells us then is that it's dependent on Wnt autocrine signaling. So since we're interested in Wnt inhibitors in the lab, we decided to treat with several Wnt pathway inhibitors. First was C59, which is a commercially available porcupine inhibitor. And you can see with the untreated versus the treated, with 1 micromolar of drug, the tumors or the organoids are much smaller, and there's fewer of them, and with 10 micromolar almost gone. ETC 159 is a clinically available porcupine inhibitor that I'm working with in the lab. And again, we see similar results. ICG 001 is basically a beta-catenin inhibitor, and it's less dramatic, but we do see similar results, that we're able to take a Wnt-independent tumor and treat it with these Wnt pathway inhibitors, and the growth is much less. So just real quick, so the specific aims of the grant, right, were to subclassify all these based on their Wnt dependency, treat them with or validate the Wnt dependency or independency with porcupine and other Wnt inhibitors, and then assess the drug responses in these organoids with standard of care agents, gemcitabine, paclitaxel, Braxane, and then the ones that showed promise move into in vivo models to validate. Long-term goals obviously would be more personalized medicine approach where we could actually use the existing bank and do all these drug screens and kind of look for any genetic or transcriptional markers that would lead to potential drug responses. But, an ideal situation would be a more personalized drug approach where we would take the patient's tumor, a biopsy, grow it as an organoid, do the screen, and we'd be able to potentially treat the patient with what we find. Long-term pipe dream goal. So not only are we able to assess the Wnt dependency, we also looked at EGF dependency. This is the same Panc 269 tumor. Here it is in complete media. Here it is without EGF. And it grows just fine. So what this tells us is that it's independent of EGF as well, potentially because of the KRAS mutant. So this was more of a validation for us that this tumor does not need exogenous EGF to grow. And so what we were able to determine then is Panc 269, since it makes its own, or is independent of Wnt, needs our spondin, is classified in this group down here based on this Sano paper. And so, again, the more Wnt independent or spondin independent the tumor is, the worse it is, and it's more potential for metastasis. So the reason these are important is more and more people are looking at organoids for drug treatment. So this came out last year in Cancer Discovery from Tubison's group. And so basically they took a very large panel of pancreatic cancer organoids, looked at transcriptional information, genetic mutational information, and therapeutic information to try to figure out if there's any correlation between all three of those, so that way they could advise better treatment strategies for patients. Other folks are looking at pancreatic cancer organoids in a more immunotherapy type. Well, since immunotherapy is so prevalent now and pancreatic cancer does not respond at all, it's a very cold tumor, a lot of people are using these organoids now to see if they can predict responses to immunotherapy. So these two papers came out very close to each other. This is from the Netherlands, where they're basically using PBMCs from the same patient where the tumor came from, and looking at responses. And this is, Hans Cleaver's involved in this. He's the king of colorectal cancer organoids. And then Calvin Kuo at Stanford is also looking at kind of trying to recapitulate the tumor microenvironment, including immune cells, to look for responses. So the last slide is basically what's been developed so far with Peter in the core. Again, we're a GI lab, so we're looking mostly at GI cancers. We have a fairly large panel of colorectal cancer models and pancreatic cancer models. When possible, we'll try to collect normal tissue as well. And in some instances, we get from the same patient, the normal colon, the primary colon tumor, normal liver, and the liver met. All these came from the same patient at the same time of surgery. The benefit of going this route also with the organoid core is there's a lot of interest from some oncologists in cholangiocarcinoma. We were able to create one as a PDX model, and that is it. So what we're attempting to do now is to take the tumor directly from the patient, give it to Peter, let him create the organoid, then take the organoid and inject it into the mouse to see if we can enrich or increase our chances of getting these cholangiotumors to grow. Peter's also working with other groups in endocrinology, Maggie Weirman to try to make salivary gland tumors, and Katja, who I'm not going to try to pronounce her last name, making adrenal cancer. So these are both tumors that have a hard time growing in mice, but it's a good model using the organoids. And that's it. Here's yours. Oh, sorry.
Definitely do questions for all three. So just, you have a question for anyone, please shout out if you want to answer.
The pancreatic tumors tend to be mostly fibroblasts. How do you think that modeling drug screening is going to be affected by that? How are you going to address that?
Yeah, so for the tumors that we take out of the mouse, it's all mouse stroma. So we basically what we're doing is getting rid of the stroma, and we just have the tumor. For the tumors that we can get directly out of the patient, Peter's actually purifying out the fibroblasts and getting them to grow as well. And then we also are purifying the tumor out. And so we could potentially do co-culture experiments where the stroma is reincorporated in. And the paper that Calvin Kuo had out in Cell, he was looking more at immune response, but he's basically doing the same thing. He's trying to recreate that tumor microenvironment with cancer-associated fibroblasts, et cetera, to try to model that better. So that there is possibilities for doing that.
So when you show your model of all the tissue that
Yeah, right. So there's nothing going on campus except for in ophthalmology, your ocular models. And so that was actually a little bit of my motivation because I would like to get into the breeding organoid business myself, and just kind of looking around and going, well, how do I do it? And so I've actually got a group of people together, and so we're talking about getting organized to put a plan together to work together collaboratively. So that is definitely actually is. We did, a student from one of my program went to a lab, a brain organoid lab, to learn how to do it. Unfortunately, she's graduating within the next month or so, but at least we got a little taste of it.
So I'll talk from the 80-90% success rate for the pancreatic cancer to grow the tumoroid. So we have another bit on growth factor disease, your organoid disease.
Yeah, so in the medium, it has, it's Wnt, noggin, correspondent, VGF, and I'm trying to remember. There's a P38 inhibitor and a TGF-beta inhibitor to inhibit differentiation.
Okay.
So it's very defined media.
Yeah.
I was just asking along with that. It sounds like you wanted to look at pediatric involvement since there's going to be a one
Would like to repeat the question.
So the question is whether there is an effort to expand this to more pediatric tumors, and I haven't discussed that with anyone, so sort of open to it.
So go back to what Scott had brought up, and sort of, can you generate more complex organoids? I mean, I know you mentioned you can incorporate in the fibroblasts, but for example, we know that the macrophages are hugely abundant in tissues. And if you incorporate them in, do they incorporate in somewhat the same manner that they would incorporate into a normal tissue? This is the anyone.
Yeah, I don't really have any good knowledge of that. I just have so again, with respect to the neural tissue, and people are introducing microglia, and amazingly enough, you would think that that alone would just activate them, right? But I think people will feel like they're having success with that sort of approach. For the neural stuff, it is black box. Everything is black box. And so one of the big challenges is how do you do this in a reproducible manner? And so there's a huge effort right now with people just trying to figure out the best way to grow these things so they get something that looks fairly uniform from one organoid to another, from one well to another. And this is one of the things that's kind of kept me back a little bit because I don't want to spend my time optimizing things. But there has been, I feel like in just monitoring the field, that there has been quite a bit of progress very recently, and I think people are pretty optimistic. There's been a series, again, from the brain stuff, there's been a series of papers here very recently, and with dramatic improvements.
Are the normal tissues more challenging to create an organoid out of? And is there anything generic about how one would do that, or are those also successful?
I think the limitation of the normal tissue, I think it's possible. People do it as long as you have your stem cells in it. So if you have like fully differentiated cells, of course, it may be really not that easy. So I think we're kind of going back to the availability of like sufficient number of cells with stem cell properties in order for them to differentiate into appropriate structures.
Hans Kleber's bit.
Yes.
They're from normal tissue.
Right.
Amazing how much they look like a little guts, epithelial monolayer and stuff.
But that's kind of where we go with this stem cells. So he uses these kind of the crypt-like structures, again, with the stem cells, which, of course, you can get. And that's what helps you with the organoids. Yes.
So I'm curious about your data on differentiation of your stem cells in the previous cells. So how long can you keep those differentiated cells in culture? Or do you have to go through the whole differentiation process every time you want to do?
Okay, I think it's more like cell line dependent. So we actually differentiate into many cell types. And for example, endothelial cells are not that bad. We can still passage them. Like, for example, these cells I showed you, they were passage 3 already, and they still look fine. I would say we can easily passage them to passage 7, 8, but then they senesce. So we also do keratinocytes, and we can passage them for quite some time, but then again, I would say they for some reason, senesce a little bit earlier than what you would expect from the normal tissue. So I think there is still some limitations in differentiation protocol. And in fact, when I mentioned that as long as differentiation protocol is available, there are technically all of the protocols are available, but I believe most of them still require some improvement so we really can get very authentic cells at the end.